Mixing human DNA with fish DNA to enable underwater breathing or other aquatic adaptations is purely speculative and not feasible with current technology or scientific understanding. Here are the key points. 1. Genetic compatibility. Humans and fish have vastly different genetic codes and biological structures. Their adaptations to underwater life, such as gills and the ability to extract oxygen from water, are complex and involve numerous genetic and physiological factors that cannot be simply transferred between species. 2. Ethical and legal issues. Genetic modification of humans, especially using genes from non-human organisms like fish, raises significant ethical concerns. It would likely be subject to strict regulations and scrutiny from bioethicists and regulatory bodies. 3. Scientific feasibility. Current genetic engineering technologies, such as CRISPR-Cas9, are primarily used for editing specific genes within the same species or closely related species. Introducing fish DNA into human cells would be extremely challenging due to differences in genetic structure and regulatory mechanisms. 4. Biological barriers. Even if it were hypothetically possible to introduce fish genes into human DNA, ensuring those genes function properly in human cells and tissues, especially in a way that confers underwater adaptation, is highly unlikely given our current understanding of genetics and physiology. In conclusion, while genetic engineering and underwater adaptations in nature are fascinating topics, the idea of mixing human and fish DNA to enable underwater breathing is firmly rooted in science fiction rather than scientific reality.